Okay, so we are starting again. Okay, oxidation of fatty acids. We are starting with the oxidation of fatty acids. Let's start with oxidation of fatty acids. Now, when I say oxidation of the fatty acids, when I say oxidation of the fatty acids, remember there are different types of fatty acids and their metabolism will be different. Different types of fatty acid will be there and their metabolism can be different. For example, we have what is known as the alpha oxidation. We have what is known as the beta oxidation. We have what is known as the omega oxidation. So these are the different types of oxidation which can be seen in the lipids: the alpha oxidation, the beta oxidation, and the omega oxidation. Alpha oxidation is seen where? Anyone? Alpha oxidation is seen where? It is seen in the peroxisomes. Beta oxidation is seen in the mitochondria. The omega oxidation is seen in the endoplasmic reticulum. It is seen in the endoplasmic reticulum. In addition, in the beta oxidation, the metabolism of small chain, medium chain, and the long chain fatty acid occurs. Okay, the very long chain of fatty acid are also metabolized in the peroxisomes by what we call by beta oxidation like process. By beta oxidation like process. In case of alpha oxidation in peroxisomes, this is the metabolism of the branched fatty acids, where branching is typically at the beta position. So this is the branch in fatty acid metabolism occurring in the peroxisome known as the alpha oxidation. Then we have the beta oxidation and we have the omega oxidation. Omega oxidation is a very minor pathway. I'm just listing it for the record. It is a very minor pathway. So these are the three different mechanisms by which the oxidation can occur. The three different mechanisms by which the oxidation can occur. But what is important and common to all the pathways, that is entry into the compartment requires activation. Entry into any of the compartment requires activation. Entry into any of the compartment requires activation, and the enzyme doing this activation, the acyl coenzyme A synthetase, is therefore must be present. The acyl coenzyme synthetase therefore must be present in all the compartments. This is one very important point to remember. Entry inside the compartment for any oxidation requires activation, and activation is done by the acyl coenzyme synthetase. Therefore, it must be present in all compartments. Okay, it is present in all the compartments. Okay, so the most common metabolism is the beta oxidation, which is occurring for the small chain, medium chain, long chain. So let's take the example of beta oxidation first, and then we'll mention the differences in the alpha and the omega oxidation. So beta oxidation occurs in the mitochondria. We have already said the beta oxidation occurs in the mitochondria. Activation required outside the mitochondria done by the acyl coenzyme synthetase. We already said it is present in all the different compartments, endoplasmic reticulum, the peroxisome, and the mitochondria. In this process, 2 ATP is required. 2 ATP is required, and coenzyme A is added. 2 ATP are required, and coenzyme A is added. Once the activation occurs, then by different mechanisms, the fatty acid can be transported inside the compartment. For mitochondria, this transport depends on the carnitine. So let's see the transport of fatty acid via the carnitine. Once activation is done, the activation is followed by transport, typically by carnitine. Please note the transport is not required for small chain and medium chain fatty acid. They can cross the membrane on their own because of their hydrophobic nature, the lipophilic nature. They're able to cross the membrane on their own, the small chain and the medium chain fatty acid. So basically the transport by carnitine is required by the long chain fatty acid. So here we can see both the things, the activation as well as the transport. The activation as well as the transport can be seen here. Let's take a look. First, okay, better, much better. Okay, first the activation. As you can see, the activation by the acyl coenzyme synthetase. The activation by the acyl coenzyme synthetase. The free fatty acid is converted to the acyl coenzyme. The free fatty acid is converted to the acyl coenzyme A. In the process, ATP has been converted into AMP, and we say that this is 2 ATP equivalent. We've already said that is 2 ATP equivalent. 2 ATP equivalent are consumed in activation of the fatty acid. The activated form is the acyl coenzyme A. The activated form is acyl coenzyme A. The acyl coenzyme A will then attach to carnitine to form the acyl carnitine. The enzyme catalyzing the reaction is the carnitine palmitoyl transferase 1. More correct name should be the carnitine acyl transferase. Why I'm saying this is the more correct name, but because it is not necessary that always the palmitic acid will be there. Palmitic acid is the long chain fatty acid with carbon 16, but it is not necessary that always the substrate will be the palmitic acid. It can be the other fatty acid also. For example, the carbon 18 fatty acid, the stearic acid can also be there, and still the enzyme will act. So the carnitine acyl transferase 1 is the better answer. We use the term palmitol just to avoid confusion with the second molecule. Okay. So the acyl carnitine is formed by the action of carnitine palmitoyl transferase 1. This acyl carnitine is then transferred into the mitochondria in exchange for a carnitine. Carnitine goes out and the acyl carnitine will go in by the enzyme carnitine, acyl carnitine translocase. If you do the mnemonic for this short form, then again it will become CAT. So just to avoid the confusion between these two, we use the term palmitol. Otherwise, the correct name should be the carnitine acyl transferase 1 only. So what has happened? The acyl carnitine has come inside the matrix by the carnitine acyl carnitine translocase. After crossing the inner mitochondrial membrane, it has now reached the matrix. It has reached the matrix of the mitochondria. Inside the matrix, the acyl carnitine is converted back to acyl coenzyme A. You can see it is converted back to acyl coenzyme A by the action of carnitine palmitoyl transferase 2. It is the isoenzyme of the first enzyme. It will catalyze the backward reaction so that acyl coenzyme A is formed, which can now be used for beta oxidation. The carnitine is used for exchanging another molecule of acyl carnitine to bring it inside the matrix of the mitochondria. This is how the transport for the long chain fatty acids particularly is occurring, and the small chain fatty acid, medium chain fatty acid may use this pathway, they may not use this pathway, but the very long chain fatty acid cannot enter. That's why the very long chain fatty acid will go to the peroxisome. It is difficult for the very long chain fatty acid to enter by this mechanism, so they go to peroxisome where they will be metabolized. So now the acyl coenzyme A is available for beta oxidation. This was the transport part. We have completed the transport. Now let us go to the beta oxidation proper. Let us go to the beta oxidation proper. The beta oxidation is a cyclical process. The beta oxidation is a cyclical process. All right. So what happens in this cyclical process? After every round, the size of the fatty acid decreases by 2 carbon. For example, if you start with, let's say, the carbon 16 fatty acid, after one round of beta oxidation, the carbon 16 fatty acid will become the carbon 14 fatty acid, then it will become the carbon 12 fatty acid, then it will become the carbon 10 fatty acid, the carbon 8 fatty acid, carbon 6, carbon 4, and finally the carbon 2 fatty acid. So every time we have a round of beta oxidation, the size of the fatty acid will keep decreasing. Every time you have one round of beta oxidation, this is how the sizes will keep changing. So what happens after one round of beta oxidation? What happens after one round of beta oxidation? One round of beta oxidation generates the following: one acetyl coenzyme A. The two carbon which have been cleaved are released in the form of acetyl coenzyme A. The two carbon which have been cleaved are released in the form of acetyl coenzyme A. In the process, one NADH, as well as one FADH2 are generated, as well as one FADH2 are generated. So this is what you get after one round of beta oxidation, meaning every beta oxidation cycle is going to produce the same thing. Every beta oxidation cycle is going to produce the same thing: one acetyl coenzyme A, one NADH, and one FADH2. The acetyl coenzyme A thus formed will go to the Krebs cycle, and the NADH and FADH2 will both go to the electron transport chain. Remember, this whole reaction is occurring inside the mitochondria, and the electron transport chain is also in the mitochondria, so we don't need any transporter. Now, how to determine how many acetyl coenzyme A, how many NADH, and how many FADH2 will be formed by the beta oxidation? So very simple. If the number of carbon in beta oxidation is, if we have the n carbon saturated fatty acid, if we have the n carbon saturated fatty acid, in that case the values are as follows: acetyl coenzyme A is equal to n by 2, NADH is equal to n by 2 minus 1. Similarly, FADH2 will be n by 2 minus 1. Alright, so see what is happening here in the last reaction. In the last reaction, when C2 is formed, this C2 itself is acetyl coenzyme A. So it doesn't need further breaking. It itself is the acetyl coenzyme A, so it doesn't need further breaking. That is why the number of reactions, or you can say the number of cycles, required is n by 2 minus 1. The number of cycles required is n by 2 minus 1. For acetyl coenzyme A, we have one acetyl coenzyme A here at the end, which has to be added to the final tally. Therefore, it becomes n by 2 minus 1 plus 1, giving us the value n by 2. Okay, now that you know all these values, the number of acetyl coenzyme A, the number of NADH, the number of FADH2 generated after one round of beta cycle of after the complete metabolism of the fatty acid, we have it here. So you have to tell me total energetics after complete metabolism. Complete metabolism means the carbon has been converted to CO2. Complete metabolism means the carbon has been converted to CO2. After complete metabolism of a carbon 16 saturated fatty acid, you have to tell me how many ATP will be formed. You have to tell me how many ATP will be formed. I'll give you the options: 106 ATP, 108 ATP, 129 ATP, and 131 ATP. So you have to give me the answer. Total energetics after complete metabolism of the carbon 16 saturated fatty acid. What is the correct answer? What is the correct answer? The carbon 16 saturated fatty acid is also known as the palmitic acid. The carbon 16 saturated fatty acid is also known as the palmitic acid. Quickly calculate with the given formula that I have given you and tell me the answer. What is the answer? Do the calculation and tell me the answer. What about the others? What about the others? What do you think is the correct answer? Okay. Recall acetyl coenzyme A is equal to n by 2 is equal to 16 by 2 is equal to 8. The NADH is equal to n by 2 minus 1 is equal to 16 by 2 minus 1 is equal to 7. FADH2 is again n by 2 minus 1 is equal to 16 by 2 minus 1 is equal to 7. Now we know that acetyl coenzyme A is going to give us 10 ATP. The NADH is going to give us 2.5 ATP and the FADH2 is able to give us 1.5 ATP. So here we get 80 ATP. Here we get 7 17.5 ATP and here we get 10.5 ATP. Total is 108 ATP. total is 108 ATP, but this is not the net energetics. When we say total energetics, you have to tell us about the net energetics. If you stop here, then you will be labeled as Vijay Mallya and Nirav Modi, that you took some loan and you didn't return the loan. Remember, you had borrowed some money in the beginning. 2 ATP required for activation. Don't forget that value. 2 ATP required for the activation of the fatty acid in the beginning by the acyl coenzyme synthetase. So the total energetics is 106 ATP. Net 106 ATP. So the correct answer is option A. Option A is the correct answer. Quickly tell me the answer for stearic acid, carbon 18, saturated fatty acid. Tell me the answer for this. The answer for stearic acid or the carbon 18 saturated fatty acid, if you do the complete metabolism, how many ATP? How many ATP are you going to get? I'll not give you the values here. You have to give me the answer directly. I'll not give you the options. By putting the value of n is equal to 18, by putting the value of n is equal to 18, the corresponding values are acetyl coenzyme 9, NADH 8, very good, FADH2 also 8, into 10 into 2.5 into 1.5, 90, 20, 12, 122 minus 2 is 120 ATP. So this is your answer here. The answer will be 120 ATP. Very good. 120 ATP is the correct answer. All of you have written the answer very correctly. I'm so happy to see that all of you are able to solve the question correctly. So I made you solve this so that now you remember, for palmitic acid 106 ATP, for stearic acid it is 120 ATP, and if it comes in the exam you don't need to solve it again. You can quickly mark the answer and go ahead. You can save your time. We'll spend time here and try to save the time in the exam. For palmitic acid, carbon 16 saturated fatty acid 106 ATP. For the stearic acid, carbon 18 saturated fatty acid, the answer is 120 ATP. Very good. Anyone has doubts about this? Anyone wants to ask anything? Now, if you want, you can note down the names of the enzymes, although they are not asked. The fatty acid oxidase is a complex including the four following enzymes. The fatty acid oxidase is the complex including the four following enzymes. They are generally not asked, but if you want, you can quickly note down these names. The acyl coenzyme dehydrogenase, the delta 2 enoyl coenzyme hydratase, the hydroxyacyl coenzyme dehydrogenase, and the thiolase. As you can see, the thiolase will cleave and release the acetyl coenzyme. The acetyl coenzyme is released by the action of the thiolase. So acyl coenzyme dehydrogenase, the enoyl coenzyme hydratase, hydroxyacyl coenzyme dehydrogenase, and the thiolase will carry out one round of beta oxidation. These will together carry out one round of beta oxidation. As you already mentioned, the last cyclic reaction will generate the 2-acetyl coenzyme. The last cyclic reaction will generate 2-acetyl coenzyme. Further breakdown is not required. The reactions per se are not important. Now we come to oxidation of fatty acid with an odd number of carbon atoms. See, this metabolism was for the even number of fatty acid. Now we have the metabolism of a fatty acid with odd number of carbon atom. What happens in the last step, rather than C2, you are left with the C3. You're not left with C2, you're left with C3. The C3 is the propionyl coenzyme. You are left with the propionyl coenzyme. The propionyl coenzyme A can quickly be converted to succinyl coenzyme A, which is a constituent of citric acid cycle, and thereby it can participate in the gluconeogenesis. We had mentioned the propionyl coenzyme A coming from the odd chain fatty acid can participate in the gluconeogenesis. Now this process by which the propionyl coenzyme is converted to succinyl coenzyme is important because it requires multiple vitamins, and these questions are asked. So we'll quickly take a look at the process. The propionyl coenzyme A, either from odd chain fatty acid or by conversion of propionate into the propionyl coenzyme A, is acted upon by the propionyl coenzyme A carboxylase. The enzyme which is acting here is the propionyl coenzyme A carboxylase, and we already mentioned previously for carboxylase the biotin is required. For carboxylase the biotin is required, so that the propionyl coenzyme A is first converted to the D form of methylmalonyl coenzyme A and then quickly isomerized to the L form of methylmalonyl coenzyme A by the enzyme racemase. So carboxylase will form the methylmalonyl coenzyme A. Now the methylmalonyl coenzyme A, once it is formed, has to be converted into succinyl coenzyme A by the enzyme methyl coenzyme A mutase. Methyl coenzyme A mutase will convert the methylmalonyl coenzyme A into succinyl coenzyme A. Here vitamin B12 is required. The vitamin B12 is required. Now in some individuals the B12 is deficient. In some individuals the B12 is deficient. If the B12 is deficient, this reaction will get blocked. In deficiency of B12, the reaction will get blocked. So what will happen as a result? As a result, the methylmalonyl coenzyme A will accumulate and it can appear in the urine as methylmalonyluria. Elevated methylmalonyl in the urine is an indication of deficiency of B12. So B12 deficiency can be estimated by measuring the methylmalonic acid in the blood and in the urine. Methylmalonic acid in the blood and in the urine, they can be measured to give us an indication of vitamin B12. Once succinyl coenzyme is formed, it can participate in the reactions just like the other intermediate citric acid cycle, and therefore it can go for the gluconeogenesis. So the two vitamins required here are the vitamin B7 biotin and the vitamin B12. B7 for carboxylation and B12 for conversion of methylmalonyl coenzyme A to the succinyl coenzyme A, conversion into the succinyl coenzyme A. So this is for the odd chain fatty acid. The even chain fatty acid will give us acetyl coenzyme A, no further requirement, but the odd chain fatty acid will give us the propionyl coenzyme A. Now we come to The alpha oxidation of fatty acid. Alpha oxidation of fatty acid. Remember, I said it is used for the branched fatty acid. It is used for the branch. It is used for branch fatty acid. अभी खत्म नहीं हुआ. अभी 1 घंटा लगेगा. It is used for fatty acids which show branching, and the branching is typically at the beta site. The branching is typically at the beta site. Hmm. Okay. So remember, one in alpha oxidation, one carbon will be removed. One carbon will be removed at one reaction from the carboxyl end, and we previously said that this is occurring within the peroxisomes. This is occurring within the peroxisome. In the condition known as the Refsum disease, the alpha oxidation becomes defective. In the condition known as Refsum disease, the alpha oxidation becomes defective. And in this case, the phytanic acid will start accumulating. The phytanic acid will start accumulating. Now, one very important point to note, the dietary fatty acid will not enter the CNS because of the blood-brain barrier. The dietary fatty acid cannot enter the blood-brain barrier, so phytanic acid doesn't enter into the brain. It is accumulating in the rest of the body and presenting the sinus symptom. Now there is another condition where the peroxisome is not working, and that condition is the Zellweger syndrome. In Zellweger syndrome, one of the two things happens. Either the peroxisome is not there, or if the peroxisome is there, it doesn't have the requisite enzymes. The enzymes which are required for carrying out the metabolism are totally absent. We call it the empty peroxisome. In both the conditions, the metabolism occurring in the peroxisome will become defective, but the hallmark of this feature is accumulation of very long chain fatty acid in CNS. Alright, so most of the guides will mention the accumulation of very long chain fatty acid in the CNS. What I want to mention here is this very long chain fatty acid is the fatty acid which was synthesized in the brain itself. It is part of the CNS, which is continuously recycled. It is continuously recycled, but when the peroxisome is not there, it cannot be recycled. It will start accumulating in the CNS. Alright, it will start accumulating in the CNS. This does not mean that phytanic acid will not accumulate in the rest of the body. The phytanic acid will also accumulate in the rest of the body, but this accumulation is much less in comparison to the VLCFA accumulating in the CNS. So in Zellweger syndrome, accumulation of VLCFA in the CNS is correct, but it is also associated with accumulation of the phytanic acid in the rest of the body. Accumulation of phytanic acid VLCFA in CNS and phytanic acid outside CNS. Remember, the amount of phytanic acid in comparison to VLCFA is much less. In comparison to the VLCFA, accumulation of phytanic acid is much less. So you can consider the two disorders, the Refsum and Zellweger syndrome, as the spectrum of disorder. When only one enzyme is defective, the Refsum. When the whole peroxisome is absent, then we get the Zellweger syndrome. This is the alpha oxidation of fatty acid for the branch fatty acid, where the branching is occurring at the beta site. And then we come to the omega oxidation of fatty acid. I told you that it is a minor pathway. It is a very minor pathway. It occurs from the omega carbon. Omega carbon is the carbon most distant from the carboxyl group. It occurs in the endoplasmic reticulum, we already mentioned, of liver and the kidney cells. It occurs in the endoplasmic reticulum of liver and kidney cell. Now look at the enzymes involved in the omega oxidation. the mixed function oxidase, the alcohol dehydrogenase, the aldehyde dehydrogenase. Why I'm showing you this? These are also the enzymes for alcohol metabolism. These are also the enzymes for alcohol metabolism, and all of these enzymes are inducible enzymes. So when there is regular excess consumption of alcohol, these enzymes will be present in a larger amount. They will be upregulated. Their synthesis will be upregulated. So not much about omega oxidation. Only thing is the enzymes are also the enzymes for alcohol metabolism. Okay, so this tells us about the different metabolism. Oxidation, we talked about the alpha, beta, and the omega oxidation. We already did the energetics. Now let's go on to the biosynthesis. We already talked about the degradation. Now we are talking about the biosynthesis, also known as the lipogenesis. Any questions so far? Anything you want to ask? Any doubts or any questions so far? So if you have no doubts and no queries, let's quickly go to the lipogenesis or the biosynthesis of fatty acid. See, I'll mention one very important thing. Biosynthesis of fatty acid is only a part of lipogenesis. First biosynthesis occurs and then esterification has to occur. However, most of the books present lipogenesis as the other term for biosynthesis of fatty acid. Please note this is not true. Lipogenesis will involve two steps. First, the biosynthesis of fatty acid, and then the esterification. Only once the esterification has occurred, we will say that the lipogenesis has occurred. Okay. So what we are going to discuss is not the lipogenesis. We are going to discuss the biosynthesis of fatty acid, and we are starting off with the okay. In methylmalonylurea, only the L form is released because the D form is quickly converted into the L form. D form is quickly converted into L form. So whatever is lost is the L form. It is not the D form. Only the L form of methylmalonic acid is released in the urine. It is not the D form. Okay, so let's do the checklist for fatty acid. The checklist. The exact process for biosynthesis is not required. The exact process for biosynthesis is not required. So we are doing the checklist for biosynthesis of fatty acid. Location where fatty acid occurs. Location where fatty acid occurs. Liver, kidney, brain, lung, and the lactating mammary gland. Liver, kidney, brain, lung, and the lactating mammary gland. These are the main organs where the fatty acid synthesis is greatly upregulated. The fatty acid synthesis is greatly upregulated.
पापा
Yeah.
पापा मेरे दादी अभी जो टीवी चल रही है तो मैं क्या देखूं
मोबाइल देख लो.
तो आज लाओ कर रही हूँ
हम्म.
श्योर
हम्म.
तो फिर मुझे एक चीज राइट करने में हेल्प कर दोगे फोन में
बाद में भी जाओ. क्लास चल रही है, सब देख रहे हैं आपको.
मुझे कोड बताना
हाँ, बाद में.
अभी
अभी मम्मा को बता. Please note that esterification is upregulated in adipocytes. Esterification is upregulated in adipocytes. The actual fatty acid synthesis in adipocytes is very less. The actual synthesis of fatty acid in adipocytes is very less. The esterification is upregulated in adipocytes. So adipocyte, strictly speaking, is not a place for biosynthesis of fatty acid, but it is a place for lipogenesis, where the fatty acid are being esterified with the glycerol to form the triacyl glycerol. So point number one, it is upregulated in liver, kidney, brain, lung, lactating mammary gland, and to some extent the adipocyte. The compartment is the cytoplasm. The compartment is the cytoplasm. The substrate is acetyl coenzyme A from carbohydrate source. The acetyl coenzyme A from carbohydrate source. Okay, please note the substrate is the acetyl coenzyme A from carbohydrate source. The end product is variable. The end product is variable. It depends on the requirement of the tissues. It depends on the requirement of the tissues. Most commonly, it is the palmitic acid. Most commonly, it is the palmitic acid. All right. In the lactating mammary gland, in the lactating mammary gland, it is the medium chain fatty acid. It is the medium chain fatty acid, means the carbon 8, carbon 10, and carbon 12 saturated fatty acid in the lactating mammary gland. In the lactating mammary gland. Okay. So most commonly it is the palmitic acid. So most commonly it is the palmitic acid, and in the lactating mammary gland it is the medium chain fatty acid, carbon 8, carbon 10, carbon 12 fatty acids. All right. So location where it occurs, we mentioned the compartment, we mentioned the substrate, we said acetyl coenzyme of carbohydrate source, and the end product we said it is variable, depends on the tissue. Most commonly it is the palmitic acid, and in the lactating mammary gland it is the medium chain fatty acid. The rate limiting enzyme and target for regulation are the enzyme acetyl coenzyme carboxylase. It is the first enzyme. It is the first enzyme of the fatty acid synthesis, the acetyl coenzyme carboxylase. It is the rate limiting enzyme, as well as it is the target for regulation. It is the rate limiting enzyme as well as the target for regulation. Let's look at the regulation. This part is important. The regulation is by hormones as well as by allosteric regulation. The hormonal regulation will operate via phosphorylation. The hormones will act via phosphorylation. Okay. The insulin decreases the phosphorylation. The glucagon and epinephrine will increase the phosphorylation. This means the insulin will remove the inactivation, and glucagon and epinephrine will cause the inactivation. Activation is done by citrate. We have already mentioned this when we are doing the TCA cycle. We had said the acetyl coenzyme is formed, will go towards the fatty acid synthesis. In addition, the citrate which is formed will activate the first enzyme of the fatty acid synthesis. However, if already a large amount of the long chain fatty acid is present, it will try to block the synthesis of more fatty acid. The long chain acyl coenzyme, if it is already present, it will try to block the synthesis of the new fatty acid. So acetyl coenzyme carboxylase is the rate limiting enzyme as well as the target for regulation. Now, another question which is asked is the cofactors which are required in the fatty acid synthesis. The cofactors which are required in fatty acid synthesis. The NADPH provides the reducing equivalents. The ATP will provide the energy. The manganese is the cofactor. The biotin is required for carboxylation. It is required for carboxylation, and the bicarbonate is a source of the carbon for carboxylation. The bicarbonate is a source for carbon for carboxylation. It is strictly speaking not a cofactor, rather we can call it the substrate. Rather we can call it the substrate. Okay, so these are the cofactors. However, these are listed as cofactors in your Harper. So I've mentioned them as the cofactors, but the bicarbonate should ideally be listed as the substrate. Now, first enzyme that we had seen was the acetyl coenzyme carboxylase. After that, the second enzyme which is required is not a single enzyme, rather it is a very big enzyme complex, and that is the fatty acid synthase complex. It is the fatty acid synthase complex. Why I'm showing you this? Because a few years back one question was asked where various enzymes were mentioned, and the question says which of the following enzymes is not a part of the fatty acid synthase complex. So you should be familiar with the names. You don't need to ratify them, but you should be familiar with the names, meaning when you look at the names you should be able to tell us, okay, this enzyme is a part of the fatty acid synthase complex. I'll try to make sense of this. See, the acetyl transacylase and the malonyl transacylase. Acetyl transacylase and the malonyl transacylase. The enoyl reductase and the ketoacyl reductase. Enoyl reductase, ketoacyl reductase, followed by the ketoacyl synthase. Okay, so these will help you to remember. They are similar sounding names. The hydratase and the thioesterase. So total we get 7 enzymes. Total we get the 7 enzymes along with one transporter protein. The transporter protein is the acyl carrier protein. One transporter protein which is the acyl carrier protein. Okay. Now you'll note that the fatty acid synthase complex is shown as a dimer. One fatty acid synthase complex here, the another fatty acid synthase complex here, and they are oriented in opposite manner. If you label this as 1, or let's say A, B, C, D, E, F, G, H, in this case the labeling is going in the opposite direction: A, B, C, D, E, F, GH. So you can see the two fatty acid synthase are arranged in you can say anti-parallel manner or the opposite manner. They are arranged in the opposite manner. Reason being, this fatty acid synthase is so long that the catalytic domain at one end are very far apart from the catalytic domain on the second end, and the substrate here, or you can say the product which has been formed here, is unable to efficiently reach the second set of catalytic domains. Alright, so when you bring the two proteins together, this substrate will now be recycled through the catalytic domain which is nearby. Alright, so it increases the efficiency of fatty acid synthase complex. The fatty acid synthase complex will always be functional. It will always be functional only as a dimer. It will only be functional as a dimer. It is not functional as a monomer. So this is about the fatty acid synthesis. I'll repeat here, the exact reactions per se are not important. The exact reactions per se are not important here. Okay, so that is about the fatty acid synthesis. I'll quickly recap how the acetyl coenzyme A from carbohydrate is coming in here to build the fatty acid. If you recall, from the mitochondria in the we get the pyruvate, which is coming from pyruvate dehydrogenase. It gets converted to acetyl coenzyme A. The acetyl coenzyme A will form citrate. It will combine with oxalostate and it will form the citrate. So from pyruvate we get the acetyl coenzyme A, it combines with oxalostate, forms citrate. When there is excess citrate, some of this citrate will be translocated to the extra mitochondrial compartment by the tricarboxylic acid transporter. This we had mentioned when we are doing the TCA cycle. I am repeating it again just to show how the acetyl coenzyme is coming from the carbohydrate. In the cytoplasm, this citrate will undergo cleavage by the ATP citrate lyase. The citrate lyase will cleave the citrate to give us the acetyl coenzyme A and the oxaloacetate. Alright. The acetyl coenzyme A will then become available for formation of malonyl coenzyme A and gradually to the formation of the palmitic acid. Gradually to the formation of the palmitic acid. So this acetyl coenzyme A is not coming from the fatty acid, it is coming from the carbohydrate. If we get the acetyl coenzyme A for fatty acid, from fatty acid it will become futile cycle. Okay, so this acetyl coenzyme A must come from the carbohydrate. Moreover, the hormonal influence is favorable only when excess carbohydrate is there, meaning insulin is required for the fatty acid synthesis. Insulin will be elevated only when excess carbohydrate is there. So that is about the fatty acid synthesis and the breakdown. So one important aspect we have discussed. Now we will do the checklist only for two more groups. One is the bile acid and the ketone bodies. We are only going to do the checklist. We'll do the checklist for bile acid and the ketone bodies. The reactions per se are not important, so no need to go into the details of the reaction. Okay. So bile acid, point number one, where does the synthesis occur? Where does the synthesis occur? Remember, the synthesis occurs only in the liver. The synthesis occurs only in the liver. Why will the bile acid be synthesized? What is the function of the bile acid? Function of the bile acids is the emulsification of the fats. It is the emulsification of the fat, so it is inside the liver and it is released along the biliary secretions to emulsify the fats. If the fats are not emulsified, they will not be successfully digested, and if they are not digested, they will be lost in the pieces. So the bile acid required for emulsification of the fats. Now the compartment involved in the synthesis of bile acid, the compartment involved in synthesis of bile acids are the cytoplasm plus endoplasmic reticulum plus the peroxisomes. All the three compartments are required for the synthesis of bile acids. All the three compartments are required for synthesis of bile acids. In addition, bile acid is synthesized by a major pathway and the minor pathway. The three compartments that I mentioned are required by the major pathway. The minor pathway also makes use of the mitochondria. So if you want to make an exhaustive list, then bile acid also require mitochondria, but that is required only by the minor pathway. The substrate is a cholesterol. The substrate is cholesterol, and thereby it is converted to the primary bile acids. I'll tell you the names of primary bile acids, don't worry. It will convert it to the primary bile acids. The rate limiting enzyme is the cholesterol 7 alpha hydroxylase present in The endoplasmic reticulum. The cholesterol 7 alpha hydroxylase present in the endoplasmic reticulum. The compartment endoplasmic reticulum is also denoted as the microsome. So microsomal or endoplasmic reticulum are the same terms. Microsomal or the endoplasmic reticulum are the same terms. The rate limiting enzyme as well as the target for regulation regulated by FXR. The target for regulation is also the cholesterol 7 alpha hydroxylase. It is inhibited by the farnesoid X receptor. The farnesoid X receptor is activated by one of the primary acids. So, in effect, what we find here is feedback inhibition. The regulation of the bile acid synthesis is by feedback inhibition. The regulation of fatty acid bile acid synthesis is by feedback inhibition. Okay, additional points. Which are the primary bile acids? The primary bile acids are the cholic acid and the ketodeoxycholic acid. However, they are not released from the liver as such. Before they can be released, they are conjugated with glycine and taurine. For example, cholic acid will give rise to the glycocolate and the taurocholate. They'll form the glycocolate and the taurocholate. In the intestine, under the influence of the under the influence of the bacteria, they are converted into the secondary bile acids. The secondary bile acids are the deoxycholic acid and the lithocholic acid. Even before reaching the large intestine, most of the primary bile acids are reabsorbed by the ileum. Most of them are already reabsorbed by the ileum. A very small amount will reach the large intestine, where then it will be converted to the secondary bile acid. Among the secondary bile acid, you can see the lithocholic acid is not absorbed. So a very small amount of bile acid is excreted. Most of the bile acid will undergo the enterohepatic circulation. So this is the important point to remember. Most of the bile acid undergo enterohepatic circulation, and we can interrupt this enterohepatic circulation. We can interrupt this enterohepatic circulation by uh, conjugating, or you can say sequestering bile acids. Bile acid sequestrants will quickly disrupt this enterohepatic circulation, and they will start mobilizing some of the cholesterol for synthesis. So transiently, if we give the bile acid sequestrants, if we give the bile acid sequestrants, transiently there can be some decrease in the cholesterol levels. So if you want to decrease the cholesterol level transiently, we can use the bile acid sequestrants. So these are some points to remember about bile acids. I'll repeat again. Synthesized by the liver, compartment required involved are the cytochrome, endoplasmic reticulum, peroxisome. The precursor, the substrate is the cholesterol. The end products are the primary bile acid. The rate limiting enzyme and target for regulation are the cholesterol 7 alpha hydroxylase, which is present in the endoplasmic reticulum. It is inhibited by the farnesoid X receptor, which itself is activated by the ketodeoxycholic acid. So in effect, it shows the feedback inhibition. The primary bile acids are the cholic acid and the ketodeoxycholic acid, which are conjugated with glycine and taurine before they are secreted. When they reach the large intestine, they undergo conversion to the secondary bile acid by the intestinal bacteria, the deoxycholic acid and the lithocholic acid. Majority of the primary bile acids are reabsorbed in the ileum itself, thereby showing the enterohepatic circulation. A very small amount is reaching the large intestine, from where the lithocholic acid form can be lost. Second group is the ketone bodies. Second group is the ketone bodies. Again, the details of the ketogenesis per se are not required. The reaction per se are not required. What you need is the checklist. What we need is the checklist. Now, ketogenesis occurs only in the liver. Again, just like bilysyl synthesis only occurs in the liver. So, checklist number one, liver. Compartment involved is the mitochondria. Compartment involved is the mitochondria. So in the liver, in the mitochondria, the ketogenesis will occur. Now please note the substrate for ketogenesis is acetyl coenzyme A of fatty acid origin. This is very important to remember. Why? Because as long as the glucose is there, insulin will be there, and the insulin will prevent the ketogenesis. We had said insulin inhibits the ketogenesis. So as long as insulin is there, ketogenesis will not occur. When insulin falls, the glucose will also fall. So the acetyl coenzyme then will come from fatty acid. So acetyl coenzyme of fatty acid origin is the source for ketogenesis, and the end product, the first product to be formed is the acetoacetate. The first product to be formed is the acetoacetate. So the first ketone body is the acetoacetate. Please note the first ketone body is the acetoacetate. The acetoacetate is spontaneously converted into the acetone. It is spontaneously converted into acetone, and it is enzymatically reversibly converted into beta hydroxybutyrate. So the two other ketone bodies are the acetone and the beta hydroxybutyrate. Okay, what is the role of the ketone bodies? Anyone? What is the role of the ketone bodies? The ketone bodies act as alternate fuel for large number of tissues. We had seen previously when we were doing the fed fast process. We had mentioned the ketone body are very important group of metabolic fuel. Very important group of metabolic fuel. The rate limiting enzyme and target for regulation is the HMG-CoA synthase. We also have a HMG-CoA synthase in the cytoplasm, which participates in the cholesterol synthesis. So this is the HMG-CoA synthase, which is the mitochondrial HMG-CoA synthase. I already told you that liver cannot utilize the ketone body, and I told you the enzyme required for activation is not there. That enzyme is known as thiophorase. The chemical name is succinyl coenzyme A acetoacetate coenzyme A transferase. This enzyme is absent in the liver, because of which the liver cannot utilize the ketone bodies. This enzyme is required for activation of the ketone bodies, before which they cannot be metabolized. Now, the ketone bodies currently they are tested by the Rothera's test. In the past, they were tested by what is known as the Gerhardt test. But remember, the Rothera's test and Gerhardt test identify the keto group. In the beta hydroxybutyrate, we don't have a keto group, and therefore both the test will be positive. Sorry, both the test will be negative for the beta hydroxybutyrate. The nitroprusside test as well as the ferric chloride test will be negative for the beta hydroxybutyrate. Both of them will be negative for beta hydroxybutyrate. The enzyme interconverting the beta hydroxybutyrate and acetoacetate is the beta hydroxybutyrate dehydrogenase. The enzyme is the beta hydroxybutyrate dehydrogenase. It will interconvert acetoacetate and the beta hydroxybutyrate. Okay, some additional points to keep in mind here. The conditions in which the ketone bodies are synthesized. The conditions in which the ketone bodies are synthesized. The first condition, where ketone bodies are synthesized is the starvation. If there is starvation, we had seen in the fat fast cycle, more and more of ketone bodies will be synthesized. In patients of diabetes, if insulin control is not good, then we see what is known as diabetic ketoacidosis. So it is again a case of relative starvation. The cells are not getting the glucose, so it is again perceived as starvation. And third is alcohol intoxication. In this case, what happens? The alcohol is metabolized in the liver, because of which the NADH to NAD ratio is quite high. In this case, even in the fasting state, the fresh glucose will not be formed. The gluconeogenesis does not occur, and the level of the blood glucose falls. So again, it is a case of starvation. For the other tissues, they will send the stimulus for the ketone body formation. So in alcohol intoxication also, you'll find the ketone body synthesis occurring in the body, giving rise to ketogenesis. Ketogenesis, if it is excess, will first cause ketosis. First, it will cause the ketosis, and if it is unregulated, if the ketosis is unregulated, then it will cause the ketoacidosis. Meaning, once the buffers are exhausted, chemical buffers are exhausted, then you get the ketoacidosis. Initially, you get the ketosis, and then you get the ketoacidosis. The acetone is volatile. When I say volatile, it means it can be lost from the lungs. The acetone is volatile and it can be lost from the lungs, and is responsible for the fruity odor seen in patients of ketoacidosis. Fruity odor of breath as well as urine in patients of ketoacidosis. Fruity odor of urine and breath seen in patients of ketoacidosis. So these are some points related to the ketone bodies that you must remember. Ketone bodies are an alternate metabolic fuel in the body. The ketone bodies are synthesized in the liver in the compartment mitochondria. The substrate is the acetyl coenzyme A of fatty acid origin. The first end product is the acetoacetate, which can be converted to acetone or the beta hydroxybutyrate. The rate limiting enzyme, as you can see, is the HMG CoA synthase mitochondrial. The liver cannot make use of the ketone bodies because the enzyme thiophorase, also known as succinyl coenzyme acetoacetate coenzyme transferase, is absent in the liver. The test for ketone bodies, the Rothera's test and the Gerhardt test, based on the nitroprusside and the ferric chloride test, cannot identify the beta hydroxybutyrate because they are based on keto group, and the keto group is absent in the beta hydroxybutyrate. The reaction catalyzed by the beta-hydroxybutyrate dehydrogenase is reversible. It can interconvert the acetoacetate and the beta-hydroxybutyrate. The ketone bodies levels are elevated in cases of starvation, the diabetic ketoacidosis, and the alcohol intoxication. Acetone, being volatile, can be lost from the breath. It is responsible for the fruity odor of the breath as well as of the urine. The acetone, when it is present in the urine, will cause the fruity odor of the urine. These are some of the points which you need to know about the ketone bodies. The reactions per se are not important. Anything you want to ask about the ketone bodies, the bile acids, the fatty acid synthesis, and the fatty acid oxidation? Take 2 minutes, quickly send me your questions. Then we'll do the last point of lipids, the lipoproteins. No questions? No questions? No questions? Okay. So if there are no questions, let's move on to the last topic for the day, and that is the lipoproteins. The last topic for the day is lipoproteins. What do you mean by lipoproteins? What are lipoproteins? Lipoproteins are special structures for lipid transport. See, lipids are hydrophobic, you know that. So they cannot travel on their own in the blood which is majorly made up of water. The lipids cannot travel. So they must be enclosed in a structure which hides their hydrophobic nature, and that structure is the lipoproteins. So let's take a look at the structure of the lipoproteins. The lipoproteins have two layers. The outer layer The outer layer is made up of the amphipathic molecules. You can see the phospholipid and the cholesterol. And then we have the inner layer. And then we have the inner layer which contains the non-polar groups. Okay. So what happens here? The phosphate part can interact with the water. It is polar in nature, hydrophilic. The lipid part of the phospholipid will be inside. The cholesterol, cholesterol has the alcohol group. The alcohol group is facing outwards, and therefore it is the hydrophilic. The ring structure is inside, which is the hydrophobic part. And we already know the proteins are amphipathic. So apolipoproteins can be present anywhere. They are amphipathic in nature. The phospholipid, the cholesterol, and apolipoproteins are the amphipathic molecules, and the core will be made up of the non-polar or the hydrophobic molecules, the triacyl glycerol, and the cholesterol ester. Please note, as soon as you esterify the cholesterol, you hide the alcohol group, the cholesterol will go to the core. It cannot be outside when you hide the hydroxyl group of the cholesterol. So cholesterol is outside. The cholesterol ester will be present in the core. The cholesterol ester will be present in the core. Typically the diagram is presented like this. The outer layer made up of phospholipid, the free cholesterol, and the apolipoproteins. The core is made up of the triacylglycerol and the cholesterol ester. Ester means to the hydroxyl group we are adding the fatty acid by esterification. Okay, so this is the generalized structure of the lipoprotein. Now at this point you have to first tell me which are the important lipoproteins. Which are the important lipoproteins you should know about? They are the chylomicron, the VLDL, the LDL, and the HDL. The chylomicron, the VLDL, the LDL, and HDL. Okay, so let's quickly first look at the composition of these lipoproteins. Before you go to the composition, I'll show you how they separate on the electrophoresis. Previously we used to do the ultracentrifugation, but nowadays we do the electrophoresis. So we'll see how the separation is occurring on the two systems. So when you're doing the ultracentrifugation, when you're doing the ultracentrifugation, means we are moving the tube at a very high speed, maybe 20,000, 30,000, 40,000, even 50,000 G. In that case the serum will separate into multiple layers of the lipoproteins. Four layers may be identified. One layer which is right at the top, having the lowest density. One layer will settle almost at the bottom, having the highest density. and then we have two layers somewhere in between. Because we are doing the separation based on the ultracentrifugation, obviously these fractions will be having the different density. The HDL at the bottom, the chylomicron at the top, followed by very low density lipoprotein, and then we have the low density lipoprotein. On electrophoresis, the story changes. Why? Because the ultracentrifugation pattern depends on density, but the movement in electrophoresis depends on relative protein content. So the pattern will be slightly different when you do the electrophoresis. When you do the electrophoresis, the pattern will be slightly different. If you start from here, if you start from here, most of the proteins are negatively charged, so they'll be attracted towards the positive electrode. So based on the relative protein content, these lipoproteins will again separate out in layers. One layer right at the top, one layer moves very fast and very far, and then we have two layers somewhere in between, two layers somewhere in between. The topmost layer is still the chylomicron. Sorry, I'm so sorry. The topmost layer is still the chylomicron. The bottommost layer is still the HDL, but VLDL moves further ahead than the LDL. VLDL moves further ahead than the LDL. So the HDL is the alpha lipoprotein. HDL is the alpha lipoprotein. LDL is the beta, and VLDL is the pre-beta lipoprotein. So you can see slight difference in the electrophoretic pattern of the lipoprotein from the ultra-centrifugation pattern. Reason being, the criteria for the two separation is somewhat different. Criteria for the two separation is different. Ultra-centrifugation depends on density. The electrophoresis will depend on a relative protein content. It will depend on multiple factors, not only relative protein content but also the composition, which gives the charge and therefore the force. Okay, so there is a difference in appearance for electrophoretic pattern in comparison to the ultracentrifugation pattern. The naming, which is old, is based on the ultracentrifugation pattern. So okay, let's talk about the different lipoproteins. What happened? Let's talk about the different lipoproteins now. There are few points which I want you to know about them, and I'll highlight them. What is happening? Okay. Okay, so what we'll do, we are going to take a look at the different lipoproteins. We'll take a look at the different lipoproteins, starting with chylomicron. Starting with chylomicron. First thing in this list will be from where they are synthesized. From where they are synthesized. Okay, then we will talk about Yeah, no energetics for ketogenesis. No energetics for ketogenesis. Sorry, I just saw the question. No energetics for ketogenesis. Okay, so from where are they synthesized? What is the primary function, or you can say content? What is the content or the function? Content or the function? And lastly, which are the markers, or you can say the apolipoproteins, which help us to identify them, the markers or the apolipoproteins. So we're starting with the chylomicron. The chylomicron is synthesized by the small intestine. The chylomicron is synthesized by the small intestine. The dietary triacylglycerol is packaged and released as chylomicron in the lymphatics. From lymphatics, it will go to the blood vessels, from there to the heart, and then to the rest of the body. It reaches the liver last. The content is very interesting. It is the exogenous triacylglycerol. It is the exogenous triacylglycerol, the dietary triacylglycerol, and the marker is the B48. B48, which is the incomplete protein. B48, which is the incomplete protein, is the marker for the chylomicron. The next in the list would be the chylomicron remnant. Once the chylomicron delivers most of its triglycerol, it becomes the chylomicron remnant. So it is obtained from actually chylomicron, synthesized from the chylomicron. Now, initially in the chylomicron, we have large amount of triglycerol, at which time the cholesterol does not become comparable. So in this case, we have equal amount of cholesterol and the triglycerol. Why? Because most triglycerol has already been taken up by the extrahepatic tissue. Now, when we talk about remnants, the remnants have the ApoE. ApoE means remnant. When ApoE is there, we call it the remnant. So this is about the chylomicron and chylomicron remnant. Let's go on to the VLDL now. The VLDL is synthesized by the liver. It is synthesized by the liver. Its content is endogenous triacylglycerol. Its content is endogenous triacylglycerol. The marker B100 and the ApoC series. B100 and the ApoC series, all of them are present. The VLDL, after it delivers most of its triacylglycerol to the tissues, gets converted into the IDL. IDL is also known as the VLDL remnant. It is also known as the VLDL remnant. So obviously it will say it is synthesized from VLDL. And this one, just like the chylomicron remnant, has almost equal amount of cholesterol and the triacylglycerol. And recall, when we say remnant, then it must have the ApoE. Then it must have the ApoE. The VLDL sometimes gets converted to the LDL. So LDL is also coming from VLDL, and this contains only cholesterol. Alright. So the function, the physiological role of LDL is cholesterol delivery to extrahepatic tissue. The tissues can take up the LDL and fulfill their requirement of the cholesterol. So physiological role of LDL is the cholesterol delivery to the extrahepatic tissue. The physiological role of LDL is cholesterol delivery to extrahepatic tissue, and the marker in this case is a single copy of B100. Single copy of B100 is the marker. And last, we go to HDL. HDL is synthesized by both small intestine and liver. It is responsible for the reverse of HDL, that is reverse cholesterol transport, and the marker is ApoA1. The marker is ApoA1. Obviously, it has all types of the apolipoproteins on its surface, but we say the marker is ApoA1. What is happening? It is not coming on the screen. What have I written? Okay, now it is coming. Okay, so these are some of the important points related to each of the lipoproteins that you must know about, each of the lipoproteins that you must know about. At this point, I'll talk a little bit about the apolipoproteins because this is something new for you. We'll quickly talk about the functions, you can say, or the properties of the apolipoproteins at this stage. What are the functions of the apolipoproteins? The apolipoproteins can be enzyme cofactors. They can be the enzyme cofactors. For example, the ApoC2 is the enzyme cofactor for lipoprotein lipase. It will activate the lipoprotein lipase, which will break down the triacylglycerol within the different lipoproteins. The ApoA1 is the activator for lecithin cholesterol acyl transferase. It is the activator for lecithin cholesterol acyl transferase, also known by the name LCAT. Basically, what it does, it will convert it will convert the cholesterol into the cholesterol ester. The LCAT, lecithin cholesterol acyl transferase, converts the cholesterol to the cholesterol ester. The apolipoproteins can also be enzyme inhibitors. They can also be enzyme inhibitors. So ApoA2 and ApoC3 will inhibit the lipoprotein lipase. See, the C2 is activating the lipoprotein lipase. The C3 is inhibiting the lipoprotein lipase. I repeat the HDL. Just wait. Just let me finish this. I'll go back to HDL. Don't worry. I'll go back to HDL. Let me finish this. So ApoA2 and ApoC3 are inhibitor for the lipoprotein lipase, and ApoC1 is the inhibitor of cholesterol ester transfer protein, written as CETP, cholesterol ester transfer protein. What the cholesterol ester transfer protein does? It has a dual role from the HDL. It will transfer the cholesterol ester to the VLDL in return for the triacylglycerol. This is done by the cholesterol ester transfer protein. The cholesterol ester transfer protein will transfer the cholesterol ester from HDL to VLDL and the TAG from VLDL to HDL. It is inhibited by the ApoC1. ApoC1 will inhibit the cholesterol ester transfer protein. It will inhibit the cholesterol ester transfer protein. Lastly, the apolipoproteins act as ligands for receptors. They act as ligands for receptors. The ApoB100 and ApoE are the ligand for LDL receptor. The ApoE is also the ligand for LDL receptor related protein known as LRP, and ApoA1 is the ligand for HDL receptor. ApoA1 is the ligand for the HDL receptor. Okay, so to recap, the apolipoproteins act as cofactors. The ApoC2 is the cofactor for lipoprotein lipase. The A1 is for LCAT. They act as inhibitors. ApoA2 and C3 are inhibitor of lipoprotein lipase, and ApoC1 is inhibitor of CETP, and they function as ligands for the receptors B100 and E for LDL receptor, ApoE for LRP, ApoA1 for the HDL receptor. Okay, before I go ahead, I'll quickly show you the HDL table that we had seen. HDL synthesized by both HDL synthesized by both small intestine and the liver. Synthesized by both small intestine and liver. It is responsible for the reverse cholesterol transport. It is responsible for the reverse cholesterol transport, and the marker protein is ApoA1, although it contains all apolipoproteins except the B series. Means B48 and B100 are not there. Everything else will be there. ApoA1, ApoA2, ApoA4, ApoC1, ApoC2, ApoC3, ApoD, ApoE. All of them will be there. E1, E2, E3, E4. All of them are there on the HDL except for B series. B100 and B48 are not there on HDL. Everything else is there. However, the marker is ApoA1. The marker is ApoA1. Okay, so that was about the HDL which you were requesting me to show you again. Let's go back to our slides, and now we'll talk about the transport of the different lipids via the lipoproteins, starting with the chylomicrons. So stay with me. First, try to understand, and then you can summarize in your own words. See, the dietary triacylglycerol is digested and packaged in the small intestine and released into lymphatics in the form of chylomicron. This chylomicron is called the nascent chylomicron. Why we call it the nascent chylomicron? Reason being, it does not have the ApoC2. It does not have the ApoC2. So this is the nascent chylomicron because it is not yet active. It is a nascent chylomicron because it is not yet active. So the dietary triacylglycerol taken up by the small intestine after digestion, packaged in the form of the nascent chylomicron and released in the lymphatics. We call it the nascent chylomicron because it doesn't have the ApoC2 at the moment. Remember, ApoC2 is required for the activation of the lipoprotein lipase. So as long as the ApoC2 is not there, we call it the nascent chylomicron. The nascent chylomicron will receive the ApoC2 and ApoE. It will receive the ApoC2 and ApoE from HDL. HDL is the repository for all the apolipoproteins. And once the ApoC2 is there, the nascent chylomicron becomes the active chylomicron. It becomes the active chylomicron. Okay, so this active chylomicron goes into the circulation. When it reaches the capillaries, the ApoC2 will activate the lipoprotein lipase. The ApoC2 will activate the lipoprotein lipase. The lipoprotein lipase will act on the triacylglycerol. and this triacylglycerol will split into the glycerol and the fatty acid. The fatty acid, as you can see, is taken up by the tissues. It is taken up by the extrahepatic tissues. The glycerol stays in circulation and goes back to the liver. The glycerol stays in circulation and goes back to the liver. In this way, majority of the triacylglycerol which is present in the chylomicron is taken up by the extrahepatic tissue. It becomes small in size and